what in the world does this have to do with hemoglobin and color? Well, the first thing is this. Most people are aware, just by way of common sense, that your skin color will change based on your activity level. If you get hot, the blood rushes to your extremity and it'll change the tone of your skin. That's because the red blood cells have color. And the color in the red blood cell is the hemoglobin that's present in those red blood cells. And if you bind oxygen to the hemoglobin, it looks different. The color of that looks different than if you don't bind oxygen to it. So you would know, for example, if a person is not receiving blood flow to an area because their skin tone takes on a more bluish or purplish tint. You know if somebody's really hot, they're exercising, my face gets red because the blood has rushed out to the extremity. My hands and my feet get red because it's trying to cool the blood off. There's more blood there, it changes the skin color. And that's because of the presence of hemoglobin. If a clinical person is going to take a blood sample. So I want some specifics about your blood. I'm going to do an ABG, an arterial blood gas. So most of the time, they take a vacuum tube with a needle on it, and they insert it into the radial artery in your wrist. That's the easiest one to get. So you know by palpating structures here with the radius where the radial artery is supposed to be. So you stick it in there, and when you hit the radial artery, the vacuum tube fills up with blood. But as it turns out, the radial vein is very close. And if you hit the vein, you will know, because the color of the blood is different. So the oxygenated blood has a brilliant red color. The deoxygenated venous blood has a dull, kind of purple, almost black color to it. And that's because of the color in the heme molecule. So it does change skin color, but hemoglobin has so much more to say about color than just skin. And this is how. This gland, the liver, is one of two primary locations for the destruction of hemoglobin in red blood cells. The liver and the spleen destroy red blood cells after they're worn out. And in that destruction, this gland and the spleen have to deliver breakdown products to the system that we can get rid of. Now, if you're going to get rid of this color, if you're going to get rid of the color on the hemoglobin, you have two options. The two normal options for getting rid of it are urine and feces. So getting rid of the color is that way. Getting rid of it by way of urine or feces. The liver and the spleen are processing these red blood cells. Now, you can actually impact skin color if you do not process the red blood cells properly. If the liver is damaged, an alcoholic has cirrhosis of the liver, there's some kind of hepatitis that's damaged the liver, and the liver enzymes are not functioning properly to break down this color molecule. Or there is some other condition, like a newborn, where there's a massive loss of red blood cells in a very brief moment. So when a person's born, this happens. You've got to convert all of the fetal hemoglobin into adult hemoglobin. So there's a massive destruction of red blood cells that happens. Now, if the system is overwhelmed or the system is not processing this colored molecule properly, that will also change skin color. So most newborns have a little yellow tint to their skin. And that's caused by hemoglobin because the system is not clearing the breakdown products of the hemoglobin fast enough. So how does this happen? Well, the liver has to process this stuff, and normally it conjugates it so that it makes it soluble and puts it in the bile. And then it takes that conjugated form and it puts it in the small intestine so we can get rid of it in the feces. So normally what happens is the liver takes the hemoglobin molecules in the destroyed red blood cells that are weak, so they've been around for 120 days, it's time to get rid of them. So the liver and the spleen are doing this. The liver then processes the heme groups, and it puts them into the small intestine. And then the small intestine takes those molecules and converts them again, even further, into a more solubilized form that can end up back in plasma. And much of that color here will end up going out of the body in the feces. So the brown color of feces is normally due to a molecule called urobilinogen, a breakdown product of the red blood cell. That's the color. Now, some of that stuff that's in the intestine will get taken up through the walls of the intestine and transported back to the liver again for another round. But this time it's solubilized, and so the liver can actually put this stuff into circulation. And this stuff now has an amber kind of yellowish color to it. And that stuff is small enough, the molecules are small enough so that you can put them in the urine. The kidneys can handle them. So the intestines and the kidneys and the liver are all playing a tag team role. Here is the outline that I will use with you in the spring to teach you exactly what happens to this molecule. The point for today is for you to learn the anatomy of the connections, because these are things you can never forget. So how, in fact, does color in the hemoglobin molecule move around? That is an anatomy question. How does it move around? Okay, if the liver is going to get rid of this stuff, if the liver's going to process this stuff, it's going to have to break the heme portion down, the hemoglobin portion down, into a molecule. You guys maybe recognize this one, bilirubin. Y'all heard that word before? Bilirubin? Nobody's heard that word before, okay? So you got to break this stuff down into a molecule that your body can handle. This is what your liver does. Your liver is breaking down the red blood cell. This is the colored portion of the molecule. You bind oxygen here to the iron, brilliant red. You take the oxygen off of the iron, a dull color. And that's skin color. And then the breakdown products of this can explain other skin colors, like jaundice, where the liver's not working right, yellow skin colors. All right, so what I want you to know is how you connect the system. How do you explain how this color is moving around with ducts? So I want to do this with these slides here that we will spend a lot more time in physiology talking through the details. And we're going to do digestive system in this class. And I'm going to ask you to remember this again. So you can see here that the liver here and the gallbladder are connected to the small intestine. So the important duct connection for determining where all this color is going is here. The hepatic ducts and the common hepatic connect with the common bile, and the gallbladder by way of the cystic connects here into the small intestine. So the processing of color comes from the liver by way of a duct system and from the gallbladder by way of a duct system. The gallbladder is just a storage organ. It has color too. Notice it when you first open your cats in the lab. It has a green color. I wonder where that comes from. This is all related to the breakdown of the heme groups of the red blood cells. So you start breaking down the red blood cells and you get all these variations in colors. Similar to the kind of strategy thing we're talking about with melanin. All kinds of variations depending on which version of the molecule you're talking about with all kinds of different colors. So melanin is an important color molecule. I can explain why the skin has different tones, why freckles and moles have their color, why your eyes have color, why your hair has color. Explain that with melanin. Hemoglobin also is a very important color molecule. The breakdown product of the red blood cell has color and explains a lot of different colors. Skin colors, for example. You move the hemoglobin, move the blood out to the periphery, you get red. Stop the heartbeat, blood's not moving out there, you turn blue. The liver enzymes are not processing it right, you turn yellow. You look at the color of the gallbladder, it's green. You look at the color of urine, it's yellow. You look at the color of the liver, it's this deep, dark purple color. Where is all this color coming from? Same molecule, hemoglobin of the red blood cell. And this is the important connection to explain how the color is moving around. So the breakdown products move down the common hepatic duct, hepatic and common hepatic ducts through the common bile, from the gallbladder into the cystic duct, from the small intestine back to the liver and then back into circulation, and the kidneys then can take it and turn it into this, get rid of it with this nice yellow color that we find in urine. Okay, so there's a lot of color to be said about melanin and a lot of color to be said about hemoglobin. Later, I will teach you the conjugation processes in physiology. But today, you're learning that hemoglobin is a skin color, and you're explaining now why hemoglobin determines so many other color phenomena in the human body. Melanin and hemoglobin. Questions? Silence. All right. Now, we also find, I put another picture. I thought I put that picture in here. Maybe I didn't. Maybe I got interrupted. Yes, I think I did right at the end of class. So I want to mention it, and then we won't spend but just a second here mentioning it. Carotene is also another pigment that has the unique ability to diffuse into the dermis and epidermis. Carotene is the pigment that is in carrots, that orange color. And I went, you can do this for yourself, just before class I was going to put the picture in so you could see it. Just type in carotene turns skin orange. I've actually seen this in person because there are some, everybody's got some weird diet that they want to put people on. And one of them is a carrot juice diet. And you can tell immediately if somebody is drinking a large volume of carrot juice every day, turn their skin orange. So this is another pigment that has to do with skin color, carotene. All right, so melanin, hemoglobin, carotene. Now let's talk about the sweat glands and sebaceous glands in the dermis. So there, as it turns out, there are several different types of sweat glands associated with the skin. Now, I want to start talking about nervous system with you now. And I'm going to do more of that today. Your skin chapter does not have this in it. It comes later in your text. But you need to start thinking about nervous system already. When we hit the head running on test 2, then you'll have to have this. So notice that there are several versions of sweat glands. This one by far is the most numerous. Eccrine sweat glands are found palms, soles of the feet, forehead. They are numerous all over the human body. The eccrine sweat gland is what you think of as being the gland that makes sweat. So it's a temperature regulation gland. As it turns out, the innervation, the nervous system information to the eccrine sweat glands is not the expected type of innervation. And I'll probably get close to this today. The eccrine sweat gland gets information from the autonomic nervous system. The involuntary. So we've brought this up briefly already when talking about skeletal, cardiac, and smooth muscle. So skeletal muscle gets voluntary. We think about that. Make the muscles contract. Cardiac and smooth, that's involuntary. So we don't think to ourselves, come on, heart, beat. We don't think to ourselves, come on, small intestine, do peristalsis. That happens without us having to think about it. That's autonomic nervous system. That division of the nervous system, in fact, has its influence on the eccrine sweat gland. And I'm going to teach you in this introductory material the logical, the fundamental logical arrangement of autonomic information. And in that, you're going to learn some fundamental categories about how to put it together, and then fundamental chemicals that are used in those categories. This one is an exception here, the innervation of the eccrine sweat gland. And it is really interesting, in part because the knowledge of the innervation of the eccrine sweat gland, in fact, explains why lie detection works. So the autonomic information that goes to these eccrine sweat glands can be used, this information can be used to do what we call the galvanic skin response or lie detection. I have the tools in the lab downstairs to do this with, and have done it as by way of demonstration for students many, many times. It's very effective using the knowledge of the innervation of these sweat glands. These are autonomically controlled. And as it turns out, the autonomic nervous system is controlled from areas in your brain that are also tied to human emotion. And so you can actually change involuntarily, in a way I can watch, the amount of sweat in the ducts of the eccrine sweat glands. And that changes conductance on your skin. And we can measure that. If something bothers you, or you've said something that's not true, it will change the conductance. And this is caused by the amount of liquid in these eccrine sweat glands. So it's very interesting, the innervation. More to come. The second type of sweat gland is called the apocrine sweat gland. And this word really speaks to the release technique. So apocrine and holocrine are different release techniques that I don't really make a big deal about in this class. But these guys, the apocrine sweat glands, are more sensitive to hormone levels than the eccrine sweat glands are. And they develop, the apocrine sweat glands develop in higher numbers as a person approaches puberty. And so the apocrine sweat glands dominate in the areas of the axilla and the anogenital areas. And the secretion of the apocrine sweat gland includes a more viscous content. So there are sometimes pieces of cells that are lost in the apocrine secretion, and that's why the apocrine secretion creates smells. This is why the locker room smells so bad, because that's rotting tissue that's part of the apocrine sweat gland secretion. Ceruminous glands, I find comical. Textbooks try to suggest various reasons for them. These ceruminous glands secrete a waxy material called cerumen that we find in our ears, in the external ear canal. They're modified apocrine sweat glands, and trying to determine what their real usefulness is, I think, is somewhat comical. Some textbooks describe them as meaningful because the cerumen actually is a deterrent to insects. I find that comical because anytime I'm anywhere there are insects, they always seem to find my ears. So I'm not buying. Anyway, they're interesting little sweat glands. And mammary glands are specialized sweat glands that secrete milk. And we'll look at those whenever we're looking at the chest. I'll show you the mammary glands as well. Okay. Sebaceous glands are another kind of dermal gland. These are simple alveolar glands. Alveolar just is the type of exocrine gland. It has a little lobule on the end of it. Simple alveolars connected to one duct. And these guys, as you have seen already, are connected. These simple alveolar glands are connected to hair follicles. We saw that in the dermis. And they're known, the sebum produced here is known to soften the skin. These glands are like their apocrine cousins, are stimulated by hormone levels. So the amount of sebaceous gland secretion will increase during puberty, and that is the cause of acne. This oily secretion blocking the duct of the hair follicle. So the sebum is a problem, especially as individuals pass through puberty, these sebaceous glands. Now, as it turns out, apocrine glands are also normally associated with the hair follicles. So whereas if you look at a section of skin, the eccrine glands all seem to poke out everywhere. The apocrine glands seem to poke out through the hair follicles, and the sebaceous glands tend to poke out through the hair follicles as well. All right, so this is another picture of it here to remind you. Here is an eccrine sweat gland. Right here, this little purple structure here, and you can see its duct extending out to the outer surface of the epidermis. And here is a sebaceous gland. These simple alveolar glands connected to the hair follicle. We pointed those out to you before. Now, at this point, I want to shift gears a little bit. This picture also shows you some of the nervous system stuff in the dermis. And this is, I know every year, at this point the students get a little bit tight because I don't feel like I'm quite ready for nervous system. But this is an appropriate, you have to believe me, this is an appropriate time to do it, to get introduced to the basic anatomy of the nervous system. This is the way every anatomy class begins. This is the way a gross anatomy graduate course begins. After some introductory material, it won't be long before skin and tissues are talked about, and the next introductory piece is always, in clinical anatomy books, the nervous system. Now, Merriam doesn't do it that way, but it does make sense to do it this way, because everything we'll talk about from here on will have nervous system in it. So it's good to get yourself an outline very early on as to what you're talking about. I use the dermis of the skin, and the epidermis for that matter, and the sensory stuff there to introduce nervous system to my students. So here's some things you know already. I described epithelial tissue as being avascular and innervated. I said it doesn't have blood supply, but to say it's innervated means the nervous system's there. So let me show that to you. I also told you that the dermis is vascularized and innervated. And there are a number of different kinds of innervation stuff that we find in the dermis. So you can see both of the two major types of innervation in the dermis on this picture. And I mentioned these before to you. You have these free nerve endings, the root hair plexus that surround the hair bulbs of the hair. That is why touching your skin is so sensitive. You touch the tip of the hair, and this stuff picks that up. You've been touched. Very, very sensitive. Free nerve endings. Now, there are also free nerve endings in the epidermis, which I'm going to show you. So these are unencapsulated, sort of, for lack of a better anatomy word, raw, bare nerve endings in the dermis and epidermis. But in addition to that, you have a variety of these specialized kind of touch receptors within the epidermis and dermis. Now, I showed you one of these in the epidermis we call the Merkel cells. There is some debate as to how useful they are because their numbers are not very big. But we can also see in this picture some other types of receptors. Here's one called a Meissner corpuscle right here. And here's one right here called a Pacinian corpuscle. And this picture is good because it shows you the Meissner corpuscle being close to the epidermis. That's good. That's where they normally are. And it shows you the Pacinian corpuscle being deep in the dermis. That's where they normally are. So what I'm going to do is I'm going to take these guys, these receptors, and track their information into the nervous system for you in order to teach you the basic anatomy of the nervous system. Just basic fundamental things in the nervous system. How does information come and go? All right? So notice down here in this picture from a different anatomy book, very similar to the one in Marriott, but I like it, and you'll see why. Because do you see what that is right there? That is a skin receptor. That is a Pacinian corpuscle. And this picture in the nervous system uses the skin to introduce a nervous system pathway. This is a fundamental, one of the basic sensory pathways, moving information into the nervous system. So there's your Pacinian corpuscle right there. Now you need some terminology. Here comes the vocab. The Pacinian corpuscle sends its sensory information along a spinal nerve. It sends its sensory information along a spinal nerve. There are 31 pairs of those spinal nerves along the spinal cord. 31 pairs of spinal nerves. How will you know which spinal nerve is communicating with which division of the spinal cord? Well, now that is going to be the subject as we move through the nervous system. In each muscle group that we talk about, I'm going to say, here's how you get the information out there, and this is where it comes from in the spinal cord. And I'm going to say, remember, young people, every time I'm going to do this, remember, young people, that normally the muscle that is innervated by a nerve that you now have learned will also represent the sensory piece of information that carries information back to the spinal cord, which means you don't have to learn it twice. For the most part, when I teach you the pathway to the muscle, you will have learned which nerve, which spinal nerve is carrying this information to the spinal cord, and which section of the spinal cord is receiving that information. Now,